Welcome to Ask the Expert, a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. We're going to post it on the Sugar Science YouTube website shortly after presentation. And if you have any questions, feel free to raise your hand in the context of the talk, or at the end, we'll take questions as we can. And today we have the distinct pleasure of having Dr. Emma Hamilton-Williams from the University of Queensland in Brisbane, Australia. And the title of her talk is Targeting the Gut Microbiota, a New Therapy for T1D ? And she is as I said, she's coming to us from the University of Queensland, where she's an associate professor. Her career is focusing on understanding how immune tolerance is disrupted, leading to the development of type 1 diabetes. And she received her PhD from the Australian National University in 2001. Then she trained as a postdoctoral fellow in Scripps in San Diego. And in 2012, she started her own laboratory at the University of Queensland's Diamantia Institute, founded by a career development fellowship from JDRF. That's awesome, great. And then she's currently an associate professor. Her lab's now focusing on understanding the role of the gut microbiota as a potential trigger for therapy or therapy for T1D, as well as developing an antigen-specific immunotherapy for T1D. Very hot topic right now, how the microbiota interacts in this autoimmune disease, type 1 diabetes. She's recently conducted a clinical trial of microbiome targeting dietary supplement aimed at restoring a healthy microbiome and immune tolerance with an ultimate aim of preventing type 1 diabetes. So she has at her disposal state-of-the-art protein sequencing and techniques to probe how disturbances in the gut microbiota impacts the function of the gut and the pancreas. She's using, in her lab, she's using nanoparticle technology to develop and deliver an immunotherapy that specifically tolerizes the immune cells that cause type 1, and she's working towards a first-in-human approach. So welcome. Thank you so much for joining us. And I just wanted to, say that we've, we're really interested in this approach. We've had Graciela Lorca from Miami, as well as yourself. And it just seems like this whole part of the field is really blowing up. People have a lot of interest in it.
Yeah, great. Well, thanks for the introduction. That was very kind.
And would you like to share some slides with us?
Share my slides? Yeah. Okay, let me do that. Alrighty, how's that looking?
Perfect.
Excellent. All right. Okay, so I'll get started then. So yeah, so we're here in Brisbane. It's summer and incredibly hot today. So it would be lovely to be over there in the US where it may be a bit cooler right now. So yeah, I'll just get straight into it. So as Monica introduced, I'm really interested in how the gut microbiota might be playing a role in type 1 diabetes, whether it could be in a part of the triggering of the disease process, whether it's a way that we could help to maybe reverse some of the increase in incidence that we're seeing in type 1 diabetes, or potentially something that could help to regulate glucose control after disease onset. So what's been really, characterized in a lot of studies at the moment is that there are changes in the gut bacteria associated with type 1 diabetes. And one of the most consistent findings that's reported is that there's a reduction in bacteria that produce short-chain fatty acids. So short-chain fatty acids are produced after we eat dietary fiber, and then there are particular bacteria, mostly in the colon, that ferment the dietary fiber and produce these short-chain fatty acids, and the most abundant ones are acetate, butyrate, and propionate. So these metabolites produced by the bacteria have many beneficial effects on the host. So they are there as an energy source for the epithelial cells and the intestine. They're helping to promote a healthy gut mucosal barrier, but they also can cross systemically into the body. They improve where they can improve immune function, they can reduce inflammation, they can increase regulatory T cells. And then they also have more metabolic effects on appetite, glucose uptake, and insulin resistance. So they have quite a wide range of effects. But then, yeah, the really biggest study of dysbiosis in the gut microbiota was the TEDdy study, and their main finding was that there was this reduction in bacteria and T1D in children before disease onset or after bacteria producing short-chain fatty acids are reduced. So the approach that we wanted to take then is, can we try to replace, so can we either promote the bacteria, expand the bacteria that produce the short-chain fatty acids, or can we bypass that directly and just deliver the short-chain fatty acids? So the approach is considered a prebiotic. So we're giving a food for the bacteria that we want to expand, but this fiber that we're using is also modified, so it has acetate and butyrate attached to it. So it's a resistant starch, a maize starch backbone, and then there's it's either has attached to it acetate or butyrate or both together. So this was originally trialled by my collaborator on the study, Eliana Marino, in a NOD mouse study, and they used these diets in mice, and they found that this is the black line is the normal chow, that if you fed these mice the hams diet that has both the acetate and butyrate together, they were almost completely protected from disease. This has gone a bit backwards. So this was also, so they found changes in the gut microbiota in the mice, but they also found that there was immune modulation. So after feeding of these two different diets, they found increased regulatory T cells, and they found a decrease in islet-specific effector T cell proliferation. And they also found modulation of antigen-presenting cells, in particular reduced co-stimulatory CD86 on B cells. So we wanted now to try to now progress this to human and find out, is this type of approach of a short-chain fatty acid yielding diet a potential therapy for T1D? So the idea is that this type of dietary approach is extremely safe. It's something that parents of young children, that there's a lot of interest in the field, and it's something that really could be potentially used even in young children that are at risk of T1D or at early onset. So it's, it's not something that's going to be, really having a dramatic effect on immune function. So it's quite a palatable approach from that point of view. So what we wanted to do first of all is just to find out if this would be safe to do in people with type 1 diabetes. So we did it in adults with T1D. It was a single arm trial. So the subjects took this starch as part of their normal diet for 6 weeks, and then there was a follow-up at 12 weeks. So the primary objective was just to determine the feasibility, the safety and tolerability of this diet. Was there any adverse effect on glucose control that would be an impediment, and could they take this much of the fiber in their diet without any gastrointestinal effects and the like. And then we had a range of exploratory objectives where we wanted to look at glycemic control, the short-chain fatty acid delivery, the microbiota composition, and immune tolerance. So it was safe and tolerable. So just to go over again what they did. So they took this HAMS AB resistant starch as a white powder. They added it to their food for 6 weeks, 20 grams in the morning and evening, just adding it to their normal food. In the first week, they got to ramp up the dose to try and acclimatise to it. And if they wanted to, they could drop down to a lower dose or stay on a half dose if they couldn't tolerate the 40 grams a day. So we had 21 participants that started the trial and 3 withdrew during the trial. So 1 was due to the supplement taste and the trial protocol, the blood draws. 1 prior to commencing for reason unknown, and 1 was lost to follow-up. So that left us with 18 participants that completed the study and the diet. So 17 of those stayed on the full 40 gram a day dose for the 6 weeks, and 1 dropped down to the half dose because of some mild gastrointestinal side effects that resolved on the lower dose. So that was really promising that actually it was highly feasible to take this dose of fibre over the 6 weeks. And as far as their diabetes control, there were no significant changes from baseline in the HbA1c, the insulin dose, or their glucose control, as we had them on CGMs for some of the time as well. So that was great. So it was safe and it was tolerable and feasible to take this amount of fibre in the diet. So then getting on more to what actually happened metabolically and in the microbiome. So the thing that we really wanted to know is, do we actually increase the short chain fatty acids? So excitingly, we looked in both in the stool and in the plasma. We found that acetate and butyrate and propionate actually all increase at 6 weeks, both in the stool and the plasma, and even actually at the follow-up time point of 12 weeks, so 6 weeks after they stopped taking the fibre, they actually still had elevated short chain fatty acids in a number of the participants. So that was quite interesting, that there was actually some kind of a sustained effect after they came off the diet. Some other things of interest to note, so these lines are all the individual participants, so there is variability between subjects, of course, particularly we saw in the plasma acetate. So acetate is the short chain fatty acids that tends to go systemically at much higher levels than the others. So you can see these numbers are very high here. So some subjects go very high in the plasma with the acetates, but actually some stay very flat, even though they tend to all be increasing in the stool. So that was interesting that there is some individual variability happening here. So when we look at,
if I can just interject for 1 second, yeah. Do you have any hypothesis? I mean, those are some really exciting results. Do you have any hypothesis about the acetate levels?
Yeah, so one of our hypotheses, so as far as what's happening with the plasma acetate, one possibility is it's actually maybe something about leaky gut. So we know that some people with T1D have leaky gut. So is it just that those people actually have more leaky gut and therefore it's crossing the barrier, or it's something, something maybe fundamentally about either the clearance, maybe through the liver, so it could be kind of that first pass clearance could be different, or the utilization, it's getting used a lot by the colon epithelial cells, so it could be something kind of fundamentally different about the colon. So we're investigating that at the moment using our proteomics approach, so our stool proteomics on these samples, where we can measure a lot of the epithelial gut proteins that are being produced to try and look at this. So that data is still being analysed at the moment. So yeah, we'll hopefully be able to work that out a little bit.
Yeah, look forward
and
to
also
that.
We're looking at how the microbiota may be different, or the immune, that what's maybe different between kind of maybe responders or non-responders as well, we're looking into. Yeah, so then we did metagenomics on the stool to look at the changes in the microbiota composition and also the function across time. So we found really a pretty dramatic change in the microbiota. So this is a supervised PLSDA approach. So it is supervised, but we found in our permanent ova, we have a very highly significant difference at 6 weeks. So the orange is the baseline, blue is 6 weeks, and then red is 12 weeks. So the 6 weeks is very significantly different from both baseline and 12 weeks. Baseline and 12 weeks can be separated here, but it's not significantly different between those two time points. So there's really a big shift at 6 weeks, and then mostly a return to baseline or similar to baseline at 12 weeks. And then if we look at the individual taxa that were contributing to the differences, I just need to move.
This is amazing. I mean, the B adolescentes is usually just, I've read, quite a bit that it's gone at onset. And then you've got, the recovery, and then this other bifidobacteria recovering. This is
so the bifido,
really
we're excited. So the bifido, it slightly increases at 6 weeks and then tends to drop at 12 weeks. So it's kind of like when you come off, they don't like it going away. But the biggest increase is in the parabacteroides, and that's something that's quite consistent in other studies that deliver resistant starches. The HAMS B was used before in healthy adults. So this is a common response to resistant starch, increasing parabacteroides. So then we had, some increases in some bacteroides and the changes in the bifidobacterium. We think that, this responder, non-responder may also be splitting a little bit on the bifidobacterium or some of the kind of healthy bacteria. So there were quite a lot of changes in the taxa. And then if we looked, where are we? At the functions, we saw even more changes in the function. So there were 48 metabolic pathways that were changed at 6 weeks again. So again, the 6 week change was, but at 12 weeks, the function tended to go back to baseline again. And some of the main changes were increase in glycolysis, and the fermentation increase was homolactic fermentation increases at 6 weeks, and then at 12 weeks they tend to go back to the butyrate production. The butanoate goes up at 12 weeks. And then there's carbohydrate degradation is decreased, and then there's a lot of increase in amino acid biosynthesis, and there was also B vitamins in particular, so cofactor vitamin biosynthesis increases at 6 weeks. So a lot of changes in the functional potential of the microbiota as well.
Can I just ask, did you see any changes? So some bacteria manufacture the precursors for neurotransmitters, or even like serotonin, right?
So those are those vitamin
biosynthesis,
those B vitamin biosynthesis. So they are the precursors for these, for the cofactors, vitamin B6.
And so they did a really nice recovery there.
Yeah, very much so.
That's so curious.
Yes, so the B vitamins have some interesting correlations that we'll come to in a second as well. So that's quite interesting. So it seemed that there was a bit of a switch in the function. So the bacteria are maybe utilizing the short chain fatty acids a lot at the 6 weeks, and then at 12 weeks they go back to producing it themselves again. So there's maybe a functional shift there. Okay, so then I'm an immunologist, and we really wanted to know what was happening to the immune system. So we used a mass cytometry panel to analyse the PBMCs across time in the subjects. So it was a 38 marker panel looking at major antigen presenting cells, T and B cells, inhibitory molecules, and co-stimulatory molecules, things like that. Now, curiously, it's a little bit of a different pattern now. So in the immune profile in the PBMCs, actually the biggest change was actually at 12 weeks rather than 6 weeks. So there's a bit of a change at 6 weeks, but the most significant is at 12 weeks. So potential. So we can't really say is that potentially it's a delayed response, that it takes a bit longer for the immune system to change, or it's something about the bounce back that we obviously really need a placebo-controlled trial to look at that, or as far as what that timing means. But what we found then were a lot of significant changes at 12 weeks and some changes also at 6 weeks. So we found a change in the composition of the T cells. So we tend to see a drop in the central memory and a drop in the naive, and an increase in effector memory populations across the T cells. We tend to see an increase in B cell populations overall, and a decrease in the antigen presenting cells, so monocytes and dendritic cells. And then if we look at the marker expression on those cells, then we can see some interesting patterns as well. So on the T cells, we tend to see an increase in TIGIT and an increase in CTLA4. So those are two really well-known immunoregulatory markers. So CTLA4 is often expressed on Tregs, and TIGIT on dysfunctional exhausted T cells, things like that. Yeah. But then on the antigen-presenting cells, so both on the B cells and some of the DCs, we saw a decrease in CD86 intensity, which was very exciting, because that was also something we directly found. It was directly found in the mice as well. So then that's a decrease. And
again, this is in human. These data you're showing are human.
Human PBMC, and that was seen to start at 6 weeks and then continue at 12 weeks. And then also CD86 and CTLA4 functionally interact with each other. So they are a ligand pair. So the CD86, the CTLA4 helps to outcompete for the CD86 binding so the T cells aren't as activated. So this was consistent. Maybe increasing more of a regulatory phenotype in the cells. So
I mean, it really suggests that, doesn't it?
Yeah, exactly. I mean, it's from the populations, it's a little harder to say, because we're decreasing central memory, but we're increasing effector memory. But then the Tregs also have the same pattern. So we see this change in the we don't have more Tregs, but we have a change in their phenotype. So then also in the markers here, this TIGIT is higher on the Tregs, for example. But then also on the cytotoxic T cells, so we've got our granzyme B positive, perforin positive T cells. So they're, for example, increasing CTLA4. So yeah, that's so quite, we think it's quite promising, quite exciting that we really can see actually kind of a systemic remodeling of the immune response in a direction that we would be encouraged by for type 1 diabetes.
For sure.
Yeah. So then the next thing that we wanted to do was then we didn't see to look at more at the clinical response and the glucose control. So as I mentioned before, we didn't see an overall change in the glucose control across time, but we wanted to then correlate with was the increase in short chain fatty acids or were the microbiota changes at all correlating with glucose control. So interestingly then what we found was that particularly plasma butyrate, also a bit the stool acetate really had this inverse correlation with HbA1c, insulin dose or time below CGM target range. So this is saying that when you have, for example, can see here the individual graphs, at 6 weeks the butyrate increases and then the insulin dose decreases or the HbA1c decreases. So the higher the butyrate, the lower your insulin requirement or lower your HbA1c. So, and that was quite exciting. And then also it was more time below range, so suggesting maybe that not going so low, that was correlating. The plasma acetate, which had this different pattern and the responders and non-responders doesn't correlate. So it was more the butyrate seemed to be more important or the stool levels rather than this high circulating plasma acetate. Then we could also look at the correlating either our short chain fatty acids or glucose control with all the different taxa or bacterial pathways. That gives us a very complicated heat map, I'm sorry, but the take home or the most exciting part we thought was really down the bottom in this red square here where we have Bifidobacterium longin and Adolescentis, so they positively correlate with the higher butyrate in the stool and then have this inverse relationship with the daily insulin HbA1c. So then the higher your Bifidobacterium, then the lower your daily insulin is kind of, you can see the actual correlation plots here. Then there were some of the other taxa, so the B vitamins, so biotin biosynthesis clustered in there, and one of these other pyrimidine pathways here as well. So this was really suggesting that we could see there were particular bacteria that maybe have a stronger correlation with better glucose control, that the plasma acetate has a really strong correlation with glycolysis pathway, so it has a little bit of a different, also with the Parabacteroides distal, which does also have that negative correlation with daily insulin as well. So yeah, quite exciting there to see, to give us some clues about, okay, which taxa may be the ones that we really need to find a fiber that can maybe target these ones a little bit more strongly, and what's the changes that we're seeing. So a lot of things still to investigate to follow up on this data.
Yeah, I mean, it's kind of like, this is just such a great observational study. I mean, you really see some strong observations. Like, okay, now next steps, like, what's the mechanism? What's happening?
Yes, exactly. And to really show with the placebo, it's a little bit tricky. One of the reasons we designed it as single arm, because in subjects with type 1 diabetes, if we give a placebo that's just a carbohydrate, then that could be problematic, for their glucose management to not know that if it's blinded. So it's so we don't know, for example, if I go on to my conclusions, we don't know, is it just the backbone resistant starch doing this? Do we actually need the acetate and butyrate? Certainly in the mouse study, you had to have the acetate and the butyrate as well, and just go through. So what we found, or could conclude, was that, yeah, the Hans AB fiber was safe and tolerable in subjects with established T1D, and it seemed that there's a significant shift in the composition and function of the gut microbiota following taking this fiber. It mostly returns to baseline after the diet stop, but the short chain fatty acid production actually stayed elevated even after the diet stopped. And then we could see this Bifidobacterium abundance in particular correlates with this increased stool acetate and butyrate and better glucose control. And we could see a significant remodeling of the immune system after the diet to a more regulatory phenotype. So yeah, the limitation obviously of our study is that it is a single arm study. So we don't know for sure that any of these effects are caused by the diet or just an association of being in a clinical trial, being on a study. We did diet surveys and things during the study, and no one reported changing their diet during the study, but we don't know that for sure. So we really need to do next, obviously, a placebo-controlled study. But we think our study really supports that this further investigation into delivery of short-chain fatty acids is definitely a potential. It could be something that's beneficial after diagnosis if it's really maybe helping to modulate the glucose control, that kind of fiber effect, maybe helping to prevent the going low, the time below target range. But the immune modulation side, we think really is an indication that this could be worth pursuing as far as prevention of looking at at-risk populations before disease.
Yeah, in the prodrome. I mean, yeah, let's exhaust those T cells.
Yeah, exactly.
I think I just think this is fantastic. I think it's a beautiful study. I know you keep going back to the fact it's like one arm, but I just think you've done some really quality capturing of which bacteria, which taxa, I guess you want to call it, were modulated, and what was the repercussion. I mean, to actually have people on a CGM and to be able to actually look at their physiological responses in context of this is even just a further step. So I think it's beautiful. I wondered if you guys have considered if there's a window, where this microbiome shifts during the prodrome. I mean, that's really like a lot of the studies show, like at diagnosis, there's been this shift, a dramatic, decrease in diversity of the microbiome. But like, has anyone looked at during the prodrome or when someone has one biomarker?
So they absolutely have. So certainly in the TEDdy study, they did longitudinal analysis, but they didn't really report in that there was a particular time prior to diagnosis that they see the changes in the in the function of the bacteria or the taxa. There was a small study by Metagenomics where they looked really from birth up until I looked at seroconversion and diagnosis, though it was pretty small, but there they really found that the change happened between seroconversion and diagnosis. So up to seroconversion, they were similar, and then from seroconversion to diagnosis was when they dropped in diversity or had these change in the microbiota function.
So after the first autoantibody. So that's an interesting window for you, your team to maybe explore.
Exactly. So yeah, I think we're going to have to go to new onset next, as that's, that kind of, it's obviously a lot harder practically to go straight into islet antibody positive children. But I think because it's such a safe, the idea that it is something that really could be used at that time, because it's, it is just a dietary supplement, it's a fiber, it's something.
Yeah, no, it sounds, very non-invasive. And
exactly.
I mean, how much of the starch of the hams, Sab, did you have to, add into whatever, a drink or whatever it was, the food they were eating?
Yeah, they were taking 40 grams a day, so 20 grams in the morning and 20 in the evening. So they had the option, they could add it to food, so it can be kind of mixed into, like yogurt or pasta sauce or that type of thing, or you can stir it into a drink. So they tended to actually prefer to just add it to a drink, so they just either put it in straight into water or like diet cordial or something like that, and they preferred to just try to knock it back, because adding it into food, they found it's a starch, so it kind of made it more starchy, or they thought it ruined the food somewhat to mix that into it. So yeah, that was
20 grams is not that much though for a substantial,
it's a big increase in what people were generally taking in fiber per day, but yeah. It was,
Yes.
they had that ramp up in the first week where they kind of ramp up a bit slowly, and they tended to kind of about the first 2 weeks they would have reports, getting used to the fibre, so kind of minor type gut issues that would resolve after a couple of weeks.
I wonder if, do you have any plans to do this type of study in the US, or you're going to keep it going in Australia? What are your thoughts for next steps?
Yeah, so at the moment, so I'm looking at some other starches and other options. So I think that from our study, it would really seem that bifidobacteria are maybe the most important things to try to increase. So I think if we could maybe investigate other types of starches that might be a little bit better at expanding those particular bacteria. So that we'll probably do locally, but we also then are looking at doing a new onset study, so that will be multi-centre and could have international sites as well.
Trial net.
Depending on funding, yeah, exactly. Yeah.
I would like
if we go to antibody positive, then it's most likely to be.
Yeah, no, that'd be fantastic. I would like to open it up to the audience. I see a couple of people here. If you'd like to unmute yourself.
I did want to just put up my acknowledgement slide as well.
Oh yes, sure. Sorry.
I want to thank everyone in my lab that was involved. So Bree Tillett was my postdoc that really did all the bioinformatics and the microbiome analysis and the correlations, but it was a really big team as well. So it was a collaboration that JDRF was really fun, helped in instigating this collaboration with Aliana, started it in Monash, and then Kirsty Bell ran the study, and Sonia in Sydney, and Barbara and Helen did all the mass cytometry, and then Esteban was also involved over in Brussels now, and Curtis and Kelsey helped with the bioinformatics as well, and of course the participants too. So thank you to all of those.
We're going to share that with our audience.
Happy to have
such a promising study.
Yeah.
Yeah, I mean, it really has so much promise, and I just can't wait to see what you do next with it. It's really exciting.
Yeah, we're excited, and we're still looking at some further, as I was talking about before, further analysis with the samples from here. We're looking at the proteomics to really try and ask a bit more about the gut function as well. Is something that we would really love to know if we have evidence of improved intestinal barrier function, for example.
Yeah. Oh yes. Okay. Again, I'll make a shout out to the audience. Is anyone interested in asking Dr. Hamilton Williams a question? Feel free. Oh yes, Matthew.
has a question.
I will unmute you. I think. Oh yeah. Go ahead, Matthew.
Great presentation and a really interesting study. On that last point, looking at assessing intestinal permeability, could you go into more detail on what you're planning on doing and how exactly you'll go about assessing that?
Yeah, so in this study, so we haven't obviously had any direct measurement of intestinal permeability by the kind of gold standard methods of, lactulose and ranulose, those kinds of things. So we're doing it by proteomics of stool. So we've done that before in type 1 diabetes, where we can, by measuring shotgun proteomics of all of the proteins in stool, so we have all the human proteins and the microbiota and pancreas proteins, so there's quite a diverse pool there, but we can then look at things like our mucins and epithelial adherens and things like that in the stool if they are changing. So we're using that approach. And we're trying it in some other samples we have where we have actual direct measurements of intestinal permeability to try to find out what are the best biomarkers from the stool proteins with kind of that classical intestinal permeability measurement. But we can also see things like calprotectin, those kind of inflammation markers, and neutrophil proteins that are all there in the proteome as well. So we're doing a lot of kind of networking and multi-omic analysis on that.
So on the stool proteome, looking at human proteins, presumably there'd be quite a few that are just part of the natural sort of shedding of the intestinal epithelium. How would you differentiate between that sort of natural
Yeah, and that is one of the problems of. So I think the more inflammation. So we do see a lot of kind of neutrophil type things. So there are clearly some things which are linked to more inflammatory type proteins, and so then we can see what's correlating there. But no, we never quite know. So if something is increased in stool, does that mean they're making more of it? So that if we see more mucin, is that because the mucin barrier is thicker, or is it because it's being sloughed off and shed and it's actually thinner? So yeah, it's hard we can infer it, but it's hard to know that directly in human studies, at least. So we can do it in mice and things like that. But yeah, so it's a lot about correlation networks. So there are obviously some quite clearly known inflammation markers in the IBD field, so we can kind of look at really what's clustering with those as well.
It's a great question.
Yeah, it's challenging for sure.
Well,
and,
you've made some great headway.
Yeah.
And with that in mind, with your time in mind, thank you so much for joining us. And we look forward to your next paper and hope everyone will be, following along, listening to this while we post it on our YouTube repository and reading your new and exciting paper.
Great.
Thanks again, and have a good rest of your day.